Welcome to CEC online lectures. I am Dr. Varunendra Singh Rawat, Assistant Professor in Zoology, Hindu College, University of Delhi. Today I will be dealing with the topic of evolutionary biology. When we see around us, we see a number of species and the organisms which belong to them, we tend to find that they vary in some way or another. If we consider our own family, our own brothers and sisters, we find that they differ significantly from us in morphology, in behavior and many other aspects. Now what exactly cause these variations between very closely related individuals, and what effect does it have on the fitness of the individuals, and how do natural selection acts on these variations and cause the evolution of species is something I am going to talk about today. So the outline for today's lectures would be, I'll be talking about what exactly is a phenotype, how are different variations in phenotypes created, what are different types of variations, and how does natural selection acts on these variations to bring about evolution of species and to bring about change in allele frequency of different genes. So the learning objective for this particular lecture would be, after this particular lecture you should be able to tell what exactly is a phenotype, what factors bring in variations in the phenotype, and how does natural selection acts on different variations of the phenotype and cause the evolution of a species. So to begin with, let's start with this particular slide. If we see this particular slide, we see peacock here, and we see that this particular bird has very big tail feathers which it can spread in a form of a fan. Now if we are to observe a number of peacocks together, and if they spread their fans, we might not be able to distinguish between the size of the fan, but a peahen would. So to us it might appear that all the tail feathers are equal in size of all the peacocks, all the fans are equal in size, but it would not be so. There would be certain amount of difference between the tail feathers of all the different peacocks. So what exactly we are seeing here is, basically we are seeing a phenotype, and we might not be able to distinguish between phenotypes of different species, but individuals of that particular species would be able to differentiate between them. So what exactly is a phenotype? Phenotype is something which is discernible to us. That is something which can be seen by us. Any trait which is occurring in an organism can be termed as a phenotype. Now these traits which I am talking about, which I am calling phenotype, they might appear directly to the naked eye. That means they could be apparent to the naked eye, like in the case of peacock. The feathers are very much apparent to us, so we can see them clearly. Or they might be at the molecular level. Molecular level in the sense that there might be a protein being produced, so that is also a type of phenotype, but we are not able to discern it directly with our naked eye. So we require certain specific techniques for us to see whether that particular protein is present in an individual or not. So when we talk of phenotype, we can say that phenotype is any trait of an organism which is basically a product of genotype of that organism, and it can be visible, it can be apparent, or it could require certain specific techniques for us to make out whether that particular phenotype is there in the individual or not. So some example of phenotypes in human which can be seen directly are height, weight, body shape, hair type, hair color, eye color, and a number of other aspects of the morphology of human. All these are the phenotypes which are discernible to us, which we can see directly. Now what exactly produces a phenotype? What causes a phenotype to appear in an individual? We all have been studying this since our lower school classes, since our 10th class I think, that phenotype is produced by genotype. And by this time you must be knowing what is the difference between phenotype and genotype. Phenotype is a trait which is being produced, which is observable to us, and genotype is the combination of genes which is present in an individual, which leads to production of the phenotype. Now, if a particular individual has a genotype for a specific phenotype, logically that individual should produce that phenotype. So suppose an individual has a genotype for producing blue hair, then that individual should always produce blue hair. But if we see in the real life example, then we see that in certain cases, even if the genotype is present, the degree of phenotype which is being expressed, it might vary. Now why does it vary? Because there are certain causes, certain other factors which come into play. So our basic logic is that if there is a genotype, it must produce a particular phenotype. So a genotype basically leads to production of a phenotype, but that is not always the case. So if we look at this particular slide, we can see that whenever there is a genotype, it has the potential to produce a phenotype, but it is also dependent on environmental factors. Some phenotypes can result if a genotype is there, but in many cases environmental factors can cause alteration of the phenotype and therefore bring variations in the phenotype being produced. So if you see in this particular slide, the genotype is supposed to lead to production of a phenotype, but because of influence or interference of any environmental factor, it will lead to changed production of phenotype. So phenotype can be a result of interaction between the genotype and the environment also. It's not necessary that if any individual has a genotype, he or she will produce the respective phenotype. For example, a person might be having genotype for tallness, but he or she might not be able to attain his or her full height because during the growing years that person did not receive full nutrition. So herein nutrition is an environmental factor, and because it was not proper, it was not adequate for the person, that's why that individual did not grow to his potential height, his or her potential height. So that we can see here that how environmental factor is affecting the phenotype and changing its expression. Or the individual could have suffered from a disease in the growing years, and that's why he could not grow very tall. So the disease here, the nutrient factor, all are environmental factors which can modulate or change the type of phenotype being produced. Let us take an example here. So I'm taking an example of spade foot toad, and I'm taking it from the book of Futayama and Kirkpatrick, 4th edition Evolution. So this particular example tells us that under different environmental conditions, different phenotype may result for any organism which is present in nature. So these spade foot toads, of course, they lay eggs in the freshwater bodies, ponds, small ponds or pools, and if the tadpoles which hatch, if there is a good supply of algae, then these tadpoles, they turn omnivorous and they store quite a lot of fat which helps them during their later life. But if the supply of algae is not that much, and if shrimp is much more in supply, then these tadpoles, they turn carnivorous, and they develop a very big head and a horny beak to catch the shrimp. Whatever changes are happening here, these are developmental changes and irreversible changes. So once a tadpole has become omnivorous, it cannot become carnivorous, and once it has become carnivorous, it cannot revert back to omnivorous. So herein there is a type of plasticity. Plasticity means a specific environmental factor causes the phenotype to become either carnivorous or omnivorous. So there is a certain adaptability or certain malleability in the phenotype, which depends on the type of environmental condition being present. But there can also be cases where the phenotype, dependent on environmental condition, it could be physiological and reversible. So herein the phenotypic plasticity is not irreversible; it is reversible, and it is physiological in aspect. So if we talk of light-skinned people, when they are exposed to sun for a very long duration, they become tanned. But this tanning is a reversible process. If these individuals are not exposed to sun later on, then they return to their normal skin, light skin color. So for this particular example we have seen, we can see that the phenotypic plasticity is at morphological level, But this type of plasticity can also be found at physiological or behavioral level also. So if we go into physiological aspect, we can find that let's take example of individual living in high altitude. We'll find that their physiology is quite different from individuals who are living at low altitude. So phenotypic plasticity is capable of occurring at various different levels, and it is dependent on the environmental condition which particular phenotype will get expressed. Now, another aspect another factor which can cause change in the expression of phenotype is the presence of epistatic genes. You must have done this particular part in your genetics class, wherein any particular epistatic genes, it leads to change in the expression of a different gene at different locus. So epistatic genes basically cause expression of some other gene at other locus to be altered. Let me take example of Bombay phenotype. You all are familiar with Bombay phenotype by now, the example of blood group, wherein individuals who have two recessive copies of H allele, they tend to have the blood group O only. Even if they are genotypically A or B or AB, they will always have blood group O. You all are familiar with why it happens. Why does it happen? Because specific enzyme which leads to formation of H antigen is missing in these individuals, and because of that particular antigen being missed in the RBCs, A antigen and B antigen cannot be formed. So even if the individual has a gene A for forming A antigen or gene B for forming B antigen, he or she her RBC do not represent those antigens because the basic substrate, that is H antigen, has not been formed. So individual will be phenotypically O. So the point here is that epistatic genes can cause alteration in the expression of phenotype, and the individual will not express the phenotype which he or she is supposed to express because he or she is having that particular genotype. Even when that individual is having a specific genotype, the particular phenotype related to that genotype is not expressed because some other gene is inhibiting the expression of that particular genotype. And another important factors which lead to alteration of the phenotype are epigenetic factors. Now these factors are being studied quite widely nowadays, and it has come to light that these particular factors, these are basically specific nucleotide sequences which are different from the nucleotide sequences of the gene responsible for production of a phenotype. So these nucleotide sequences, they are not within the gene which is responsible for production of phenotype. They are far apart. They might be far apart or they might be nearby, but they are not part of that gene which produces the phenotype. So what happens here is that these epigenetic factors, they cause change in the change or alteration in the DNA, or they produce RNA, which leads to alteration of the expression of the genotype. Now how do they do so? There are a number of mechanisms by which they do so, and these are DNA methylation. One of them is DNA methylation. What exactly happens in DNA methylation is methyl group gets added to nucleotides at specific location of the DNA. It usually happens in the region which are rich in C and G. CG rich regions are the ones which usually go and undergo methylation, and the C nucleotide is the one which undergoes methylation. Due to addition of methyl group, there is change in the structure of DNA, and therefore because of this change there occurs a change in the interaction of the DNA with the proteins which are needed for transcription. That is, the proteins which are required for transcribing that particular gene into RNA, they might not interact properly with the DNA if it is methylated. So if the region is methylated, if a region nearby the gene is methylated, or the promoter region is methylated, then that particular gene will not get transcribed. So this is one of the way, methylation is one of the way by which alteration in the expression of genotype can happen. Second way by which this can happen is histone modification. Now histone, as you all know, are the proteins which are required for chromatin folding, which are required for packaging DNA properly. and these proteins have a good amount of lysine and arginine residue. Now these proteins, if they are modified, then they can lead to alteration in the way in which DNA is packed, and it can cause the DNA to be converted to heterochromatin or euchromatin. Now how does it do so? It is by a process of acetylation and methylation. Now acetylation is usually associated with active chromatin, that is euchromatin usually has a region which has histones which are acetylated, whereas whenever there is deacetylation, that is acetyl group is removed from histone, then the region becomes heterochromatin. So acetylation can cause a region to become euchromatin so that it is expressed, whereas deacetylation can cause a particular region of the DNA of chromatin to become heterochromatin so that the genes which are in that particular region are not expressed at all. Methylation can also do so, but methylation has both the capabilities. Its addition at specific locus can cause activation of genes, and at some other locus it can cause deactivation of the chromatin in that particular region. The third way by which epigenetic effects come into force are RNA associated silencing. So what happens here is that genes get turned off. Genes get turned off because either the epigenetic regions, they form RNA which are complementary to the RNA being transcribed by the gene, or because of RNA interference. And if this happens, then whatever gene is required for production of a phenotype, it does not get expressed, and therefore the phenotype is not being formed. So recent research suggests that more and more regions of DNA which were earlier supposed to be non-functional, to not code for anything, they code for RNAs which are complementary to a number of regions, to a number of genes, and because they are complementary to those particular regions, they can bind with the mRNA being formed by those genes, and once that happens, a double-stranded RNA is formed which is then cleaved. So basically what happens? Expression of that particular gene is nullified. So these are all different effects by epigenetic factors which can lead to alteration of phenotype. Now this particular picture or slide beautifully sums up all the different factors which are responsible for expression of a phenotype given a specific genotype. Now we can see at the lower panel that environmental signaling, which involves pathological factors, nutrients, physiological, toxins and behavioral factors, it can lead to alteration of a phenotype being produced by a specific genotype. Also we can see on the right hand side panel that methylation, acetylation and basically DNA methylation, histone modification and effect of RNA silencing can lead to change in the expression of phenotype, and the phenotype which is being produced is different from what should have been produced given that particular genotype. Now if you look at this particular picture, this particular panel shows a large number of corn cobs, and we can see that they are different in shapes and size. Also the color of seeds is quite different in a great number of them. Even so, if we see the topmost panel from right hand side, if we see the second corn, we see that in the same corn cob there are seeds of different color, purple color and creamish color. So all these corn cobs are basically displaying variations. What are the factors here? I am not going into that. You already know what factors cause variation in the color of the seeds of the corn, but what role does variation play in evolution? I will be talking about that. So before I move forward, I should make a point here that all the sexual organisms, they display some sort of phenotypic variation. Why that is so, that I'll be covering in another lecture, but all the sexually reproducing organisms show variations in their phenotype. Saying that, it doesn't mean that organisms like prokaryotes and viruses do not show variation. Even bacteria show variation, which we have already done in our genetics and biotech classes, that there are a number of strains of bacteria even if they are belonging to the same species. Some strains might be showing resistance to specific antibiotic, while other strain might not be showing resistance to that particular antibiotic. So this is also sort of variation, which is again being brought about by presence or absence of a specific gene. Also, we tend to hear that viruses are mutating and a number of strains of viruses are getting formed every day. Like in case of coronavirus, also we tend to hear that there are a number of strains here, and it's difficult to find antibody or vaccine for this particular virus. So how is this virus forming different strains? That is by mutation of its genes, and all these strains are showing variation in those particular genes. Because of mutation, nucleotide sequence is changing and variations are developing within the genome of the virus. So basically what I want to tell here is that all the organisms, whether they are sexually reproducing, whether they are bacteria and virus, they tend to show some form of variation. Even if it is not at the morphological level, it can be at the genotypic level. So when we talk about variations, we can classify the variations as morphological, that is those which are visible to the naked eye, or physiological, that is which are apparent in physiology only, which we cannot fathom, which we cannot see, but they affect the physiology of the individuals. Or they could be at molecular level, that is change in the nucleotide sequence, variation in the nucleotide sequence of a particular gene or a number of genes. Or they could be at protein level, that is variation in the sequence of the amino acids. All these are molecular variation. Now, one very important differentiation of variation which we need to make is germinal or somatic variations. Now, these germinal or somatic variations, basically when we talk of somatic variation, these are the variations which occur in the somatic cells, whereas variations which occur in germinal cells, when we talk at molecular level, these are the ones which occur in the germ cells. So if there is a difference in genes at the germinal level, then only that difference can be passed to the next generation. If there is a difference, if there is a mutation in genes in the somatic cell, that will not be passed on to the next generation, and it will die with the individual. So only those variations at molecular level, that is at nucleotide level, which occur in the germinal cell, they can be passed on to the next generation. But those variations which occur because of mutation in the genes in the somatic cells cannot be passed on to the next generation. Now, talking about the phenotypical aspect of variation, if we try to distinguish these variations, we can talk of variations as continuous variation or discontinuous variation. Now, these continuous variations are those variations which usually are result of the additive effect of multiple genes. Now, each of these genes which produce an effect, by themselves they produce a small effect, but the cumulative effect of all these genes which lead to production of a phenotype is quite large. So if these effects are independent of one another and these small effects, if they add up, then the variation in population will show a normal distribution. Now, what exactly is the normal distribution? It is a bell-shaped curve. Normal distribution, if we see in this particular panel, we see the height of women and men for a particular population. We see that most of the individuals are average in height. That the peak or the the peak of this particular graph shows the average individual. There are very few who are less in height than the average height, and very few who are more in the height than the average height. So this particular bell-shaped curve will be found in different population. Might be the average height varies for different population. Say Asian population and American population. The height for the American men and American women will be, average height will be more than the height for Asian men and Asian women. But if we talk of that population only, in that population we'll see that the distribution of height will be a normal shaped curve or a bell-shaped curve for both men and women. So continuous variation usually show distribution which is a normal distribution. Now, these traits, continuous traits, of course they result from interaction of the alleles which they have inherited from the parent, and the genotype which occurs in these individuals, it predisposes the individual to show a particular phenotype, but environmental factors also play a part here. Apart from environmental factors, there will be other factors like epigenetic effects and the epistatic effects which will also play a part here. Now, coming to another type of variation, that is discontinuous variation. These are variation basically which show discrete phenotypes. and there is no intergradation in between. So there is no intermediate value. There are discrete phenotypes, like if we talk of blood group in human, ABO blood groups, either they are blood groups A or it is B or AB or O, there is no intermediary blood groups. These are discrete phenotypes, and the genotypes of these trait can be in most of the cases deduced directly from the phenotype. Also, if we know what is the phenotype of a particular individual, say of parent, then we can guess or we can predict what could be the phenotype and genotype of the progeny of that particular couple. Usually these phenotypes are not altered by the environmental influences. So this is how we see that there are discontinuous variation and continuous variation. And if we talk of variations in the blood group frequency in different population of humans, if we see African population, it has ABO frequency near to the world average. If we take Caucasian example, the allele A is somewhat more frequent than African or Asian population. If we take Asian population, the frequency of allele B is quite high. Whereas in Native American, the frequency of allele O is quite high and the frequency of B is almost non-existent. Whereas for Australian and Pacific Islands, the frequency of A, B and O alleles is close to world average. Now, why there is so much variation in the frequency of these population? Can you make an educated guess? Or if you are not able to make a guess, you can go back to your readings and find out why exactly it is so. Why these different populations have different frequencies of the alleles. Talking about the variation and how natural selection acts on them, I'll take an example of sickle cell anemia and malaria. Now, you all know that malaria is a disease which is quite prevalent in tropical and subtropical region, and it is caused by species of Plasmodium, different species of Plasmodium, which has a complex life cycle, and it requires to host basically humans and the mosquito. Now, in humans, these parasites live in liver and red blood cells, and of all the species, Plasmodium falciparum is the most virulent species. It was seen in some individuals that their RBCs they resembled sickles. So it was found that this particular condition is caused by an abnormality in hemoglobin due to a change in the amino acid at 6th position. The amino acid glutamic acid is replaced by valine. So individuals who have 2 copies of this particular allele, they tend to die, and those who are having one copy of this allele, mutated allele, and 1 copy of normal allele, they were normal, and those who had 2 normal alleles, of course, they were normal. Why the individuals who had 2 copies die? Because sickle cell led to resistance in flow, and this led to impairing of the microcirculation and blocking of the capillary. This impaired microcirculation further reduced the oxygen level, which resulted in severely painful sickle cell crisis, and the sickle cells got destroyed, and the frequency of destruction was much more than the normal shaped round one, and this led to decrease in oxygen carrying capacity, which led to anemia, and the individual died. Now, the test of various African population show that there is a high frequency of the mutated allele, that is up to 25% in certain population. Now, how could such a mutated allele, which leads to impairment of the function of RBC, can be found at such high frequencies? Because I have already told you that such individuals, such particular mutated allele leads to reduction in the carrying capacity of the oxygen by the hemoglobin of the mutated cell. So why this particular allele is found in such high frequency, as high as 25% in certain population, and in other population it's up to 12%. It was observed that the areas where the sickle cell allele was frequent were also the areas which had a high incidence of malaria, particularly the variety caused by Plasmodium falciparum. Now, can you make a link between these two? Individuals who are heterozygous, they are well able to survive even in the areas which show high incidences of malaria by Plasmodium falciparum, because this particular parasite cannot live or cannot thrive in the cells which have the mutated hemoglobin. So individuals who are heterozygous, they tend to survive in these regions even if they are attacked by this Plasmodium parasite. Whereas in normal cases, if there is no parasite, Plasmodium parasite, then in those population, like in the temperate regions, the frequency of this allele would be very low. So this particular allele is conferring an advantage to the populations which are living in a region where the infestation by Plasmodium falciparum is quite high. So this particular example is an example of overdominance. That is, the variation is being the genetic variation is being favored. So here we are seeing that even though the allele is not suitable, or allele is harmful to the individual, it is being selected because it is conferring an advantage in that particular population. So this is an example of overdominance, where population evolves to a stable polymorphic equilibrium. Otherwise, in normal population, this allele would have been removed altogether and the only allele would have been HPA. But herein, overdominance is basically selection of heterozygous individuals, and herein heterozygotes are being selected and the population is evolving to a stable polymorphic equilibrium where there are more than one allele being selected. That is, 2 alleles are being selected. So there are various such examples in nature which show balancing selection, wherein the population evolved to polymorphic equilibrium. Also, there are examples have been seen where there is frequency dependent balancing selection. So one type of frequency dependent selection is negative frequency dependent selection, which can lead to balancing selection and polymorphism, wherein the allele which is lower in frequency is selected, and because it is selected, then its frequency increases. So what we have a population which tends to have more and more types of allele at a particular locus. So negative frequency dependence also leads to balancing selection. Also, multiple niche polymorphism, where different genotypes are specializing on different ecological niche. This particular type of polymorphism also leads to balancing selection and evolution of polymorphic equilibrium. But there are cases where heterozygous are not selected, and this lead to underdominance, where heterozygotes have lowest fitness, and because of the elimination of heterozygous, the variation is lost, and this particular population has less and less variation, and only one type of allele will be found in these particular population. And another case where this happens is positive frequency dependence, wherein the allele which is highest in number, or the phenotype which is most prevalent in that particular region is selected. Any new phenotype, any new allele which is introduced into the population, or any new phenotype which is formed in the population is removed. Therefore, this particular case shows that it leads to prevalence of only one type of allele and only one type of phenotype, like as seen in Heliconius erato, where there are a number of geographic races present, but they all have different color pattern. If you take individuals from one race and mix it with other race, that individual will be removed from the population. Only one type of pattern is followed, because that particular individual will be very clearly recognized by the predator. Because all these Heliconius are distasteful and they are poisonous. But if a new individual is introduced, the predator might think that this individual is tasty, or it is not toxic, and that predator will focus on that particular new individual and remove that individual from the population. So this type of positive frequency dependent selection leads to removal of variation from the population. So these examples show you how variation is acted upon by natural selection, how variation can increase within the population when natural selection acts on it, and how variation can be decreased when natural selection act upon it, both at the allelic level, that is at gene level, and at phenotypic level. So that is all for today. Thank you.